Hello everyone, I am Dr. Ujjwal, Assistant Professor, Department of Botany, Rampurhat College, West Bengal. My selected topic is Dependency of crops on pollinators and pollination deficit, considering the influence of various reproductive traits. Pollination is a vital ecosystem service required for plant reproduction. About 80% of world food crops rely on pollinators. Crop plants depends on different pollinators like cucurbita, mostly on honeybees, bitter gourd on helictas, Raphanus on stingless bees, pigeon pea on leafcutter bee. World pollinators are decreasing for many reasons, including loss of habitat, use of pesticides, and climate change. As a consequence, there may be risks of pollination deficit, compromising crop yield and quality. Therefore, the present work was designed to assess the pollinator dependency and pollination deficit of crops. In addition, we also estimate the reproductive traits how influence on these two parameters. Our study conducted in 3 districts of West Bengal, mostly in rural habitats, prevailing agricultural activities. We selected 17 crops available on the study region. We estimate reproductive success fruit set in 3 pollination systems: open pollination, pollinator exclusion, and supplementary pollination system. Then we calculate pollination dependency index on basis of reproductive success in pollinator exclusion treatment and reproductive success in supplementary pollination system. We also estimate a coefficient of pollination deficit considering reproductive success in open pollination and reproductive success in supplementary pollination system. We collected data on reproductive aspects of the selected crops like longevity of flower, pollen production, ovule production, stigma receptivity, and amount of visits received a flower. For normality test, we conducted Shapiro wheel test. For heterogeneous data, we used non-parametric Kruskal-Wallis test, and normally distributed data we used one-way ANOVA. From our experiment, we showed that fruit set of different crops varied among the 3 treatments based on pollinator dependency index. Member of cucurbitaceae shows obligatory dependence on biotic pollinator. Fruit plants of Rutaceae and members of Brassicaceae also showed higher dependency on biotic pollinators. Some crops like Cajanus cajan, Phoeniculum vulgare showed moderate dependency on pollinators. Only a few plants like Lablab purpureus shows little dependency on pollinator. Here table 1 showing the dependency of pollinators and their pollination deficits. Member of Cucurbitaceae shows obligatory dependency IDCP value 1. Among the selected crop, citrus shows higher pollen transfer limitation. Few plants, Cajanus cajan, Cucumis sativus, showed little pollination deficit. On other hand, a few plants like Brassica do not show significant pollen transfer limitation in nature, open pollination system. Pollination deficit varied according to crop species and geographical region, mainly depending on the abundance of effective pollinator and number of visits received the flowers. Reproductive traits of the selected crop also varied. Higher longevity for flower recorded for Rutaceae, Solanaceae. Higher pollen yield recorded for Cucurbitaceae, Solanaceae. Less number of ovule found in Apiaceae, Fabaceae. Pollen ovule ratio remained higher in Cajanus cajan, Solanum lycopersicum. Higher number of visits recorded for Phoeniculum, Brassica, Raphanus and Nigella. Here table 2 showing different reproductive traits of the selected crops. The value of correlation coefficient shows that pollinator dependency of the crops negatively correlated with flower longevity, pollen yield, ovule number, pollen ovule ratio. Coefficient of pollination deficit has a medium correlation with stigma receptivity and a low correlation with flower longevity and pollen ovule ratio. Table 3 showing value of correlation coefficient among different traits. In conclusion, members of Cucurbitaceae are obligatory dependent on biotic pollinator. Members of Brassicaceae rutaceae highly reliant on biotic pollinator. A few crops, Lablab purpureus, Nigella sativa, are less dependent on pollinator. In open field condition, most crops have little pollination deficit. Some are without pollen transfer limitation, and only a few plant crops, citrus, lemon, citrus maxima, showed higher pollination deficit. Pollinator dependency index is negatively influenced by the pollen ovule ratio, which also negatively affects the pollination deficit of the crops. The current study will be helpful in understanding and mitigating the consequences of pollinator declines. It also helps select crops to provide supplementary pollination services to enhance crop yield. These are some references. I am thankful to the organizer. Thank you.